Hi, I'm Sue Shearer, author of Guide to the Human Genome. In this demo, I'd like to take you through some of the features of the guide. In many ways, Guide to the Human Genome is much like a conventional textbook. It has a table of contents divided into chapters and sections. But in addition to its more conventional aspects, the guide has a database associated with it that provides many different ways to find the information you might be looking for. Instead of an index, the guide has a search facility. In this example, what I'm going to do is enter the word aldolase, name of the enzyme in glycolysis. And as you can see, when the search is done, the results appear in a frame at the bottom of the page. And not surprisingly, the most number of references to aldolase are found in glycolysis. Rather than searching the text of the guide, it's possible to search instead the database of human proteins that's associated with it. In this example, what I'm going to do is enter the beginning of a human gene name, and when the results appear at the frame at the bottom, you can see the entries for the 3 human aldolases. There are multiple entries for aldolase A because the database is built from the RefSeq set of human proteins, and if there are multiple transcripts in the RefSeq database, each one yields a distinct protein entry, whether the proteins encoded are identical or whether they're different isoforms. Instead of searching the gene names, it's possible to search the NCBI descriptions of the genes. Again, I'm going to enter aldolase, and when the results appear at the bottom, you'll see not only the human aldolases, but also some other genes where aldolase is part of the description, including transaldolase in the pentose phosphate pathway. Selecting the links in the results from the gene searches takes you into the database part of the guide. Here I'm showing an entry for aldolase A. At the top of the page on the right is a link that takes you to the matching NCBI RefSeq entry, which then provides vast amounts of additional information and links to other NCBI resources. Just below that at the right, you can see the link that will take you into the glycolysis section of the book where aldolase is described. Some proteins are found in many sections of the guide. Scrolling down the page, you see information about protein composition, a variety of links to do comparative genomics analyses, and finally at the bottom, a section about related human proteins. There are many additional links on each database page. Larger versions of both figures are available by selecting them. Detailed information about matching proteins in other species is provided by one of the links at the right. Similarly, one can get the details about other matching human proteins by selecting the BLAST-P results near the bottom. If you scroll through the BLAST-P results, you'll see that there are yet additional links. Instead of taking you to NCBI, these links take you to the database pages in the guide for those matching proteins. Perhaps the most interesting way to navigate the guide is to start with a protein sequence. Here I'm going to enter a predicted protein sequence from C. elegans. As you'll see when the results appear, this sequence is related to the human aldolases. If you scroll down the search results, again there will be links in the results not to NCBI, but to the database pages in the guide for the human proteins that match. The heart of the guide are the sections that describe human biochemical pathways, protein families, cellular processes, and other functions of interest. Here I'm showing the section on glycolysis. You can see there are figures, tables, numerous links to the database pages of the guide, and at the bottom a section entitled Notes and References. This section will include technical details about the figures and links to NCBI for proteins and other species and for human nucleic acid sequences. Each chapter of the guide ends with a section that provides additional readings. These include textbooks, review articles, and individual research papers, particularly on subjects that might be difficult to locate or I found to be of particular interest. I hope this brief tour will help you get started using my guide to the human genome. Thanks for listening.